Bruce, you are up. Thank you so much. Bruce Damer, also from the University of California, Santa Barbara. I'm sorry, Santa Cruz. Always get those backwards.
Yeah, if we were in Santa Barbara, we would be surfers right now.
No, right.
Right. I want to thank Michael and the organizers of Columbia Institute of Astrobiology. It's sort of poignant that we're doing this today because the ISIL meeting, which had to get rescheduled, was going to be in Ecuador, in Quito, this year, and bring astrobiology from ISIL to the Southern Hemisphere the first time, but obviously we didn't have it. So it's great to sort of have a presence in South America. So I'm taking off where Dave left off. Several speakers today have talked about Darwin's warm little pond intuition, and as we heard, the BBC ran a major story yesterday. Was Charles Darwin right all along? And that's sort of got it into the news. And everyone's familiar with the statement, the first part of the statement, if, and what a big if, we could conceive in some warm little pond with all sorts of ammonia, phosphoric salts, light heat, etc. But people don't really pay attention until now to the second part of the statement, that a protein compound was chemically formed, ready to undergo still more complex changes. Charles Darwin in 1871 was describing an away from equilibrium system. We form a polymer made with amino acids, and we allow it to become more complex. And so this is the 21st century challenge that many of the speakers are taking up. Just an amazing prescient statement in this brief letter to his friend. So I'm going to take the big picture, as Dave said, and look at the terrestrial landscape to search for the Goldilocks conditions for Darwin's warm little pond. What we think is that on any terrestrial landscape, you have the salt water at the intertidal zone starting, and then you have fresh water, distilled water evaporating from the ocean and coming down on the land, getting itself into environments like this, into hydrothermal fields in a volcanic setting. And this is where we believe that Goldilocks chemistry can set up, as proposed by Sutherland and Pounder years ago. There's a little pool in Kamchatka that Dave used and poured his reagents into. We think that life can start in the Goldilocks chemistry and over time adapt across the landscape to the more extreme environments of the ocean. As Mulkajanian reported in 2012, life really had to adapt to salty conditions later because it has complex, ion energy-driven pores to do that. It couldn't really have started in those conditions. So the landscape contains essentially a fitness landscape of gradients, which can collect, concentrate, cycle, adapt, stress, and distribute early life, even the early proto-life, and then the earliest forms of life. There's converging lines of evidence behind this from the rock record. Our colleagues at University of New South Wales 4 or 5 years ago discovered Geyserite in the Pilbara, 3.5 billion years old, discovered by Tara Jokic, a graduate student of Martin van Krenendonk. You can see it on the left, these bands, which are indicative of a splashing geyser, the oldest one discovered in the solar system, laying down material, and it's a very distinctive mineral. And in the midst of the Geyserite is clear signs of microbialites, living organisms far back, including what seemed like oxygenic bubbles preserved in silica. It's a very, very compelling discovery that rich ecosystems were present on land as far back as we can look. So the old idea that the continents were dead and that life emerged from the oceans has long been sort of put on the shelf. Here's some subsurface vein stromatolites that I found with Tara and field work, also 3.5 billion years old. This is in barite. Here's lacustrine, lakeshore stromatolites, where the microbial mat sequesters the mud and sand grains, and is through onlapping wave action gets folded up. And so this is lakeshore stromatolite at freshwater 2.7 billion years ago, and this is Shark Bay living stromatolites today. So you can see a continuity in the rock record going all the way back as far as we can look. As Dave pointed out, and several other speakers, There's evidence of organic sources from meteoritic sources, such as has been shown with the Murchison meteorite, where amino acids, carboxylic acids, and even sugars are coming in from space on these objects and in IDPs. Ben talked about a biogenic atmosphere. Also, the Europeans determined that 40% of the compounds in the dust from the comet on the Rosetta mission is organic. So this feedstock for meteoritic components, volcanic emissions, biogenic atmosphere can concentrate in pools on land, but they will be lost in the dilute bulk of the oceans. And Dave mentioned earlier doing our hot spring field work. This is from 2018. We just repeated the results in February 2020. There we are basically cycling, wet-dry cycling, this hot plate of vials of these mixtures of RNA monomers and building blocks using hot spring acidic water, wet-dry cycling them. And what you can see there is the films of the lipid of the amphiphiles clearly visible during the dry phase. There's mobility of the monomers in between those layers, and that mobility allows them to form these chains that Dave talked about earlier. It's just visible with the naked eye. And so RNA-like polymers are formed both in the laboratory simulation and in the hot spring environments. And again, we replicated this again in February with teams from Auckland and Sydney. So as Dave also pointed, that when you wet dry cycle, the protocells bud off and can encapsulate polymers. Some of them will encapsulate polymers. You can see A, B, C, D, and we're getting more and more encapsulation, in this case of DNA that is stained so we can see it. This sets up a roulette wheel. Now we have all these experiments that are going to run against stresses. So this is what we came up with, that there's a cycling selection going on in this wet-dry cycling, which goes through three what we call coupled phases. The dry phase on the left, where these layered systems allow the synthesis of the polymers to occur. The budding, and we can see an image of budding, of protocell budding happening. When you add that solution back in, it tests these protocells for structural stability. The surviving protocells that don't break up and lose their contents are concentrated down to an interactive gel phase at the bottom, where they're simply resting at the bottom of the dish or the pool. And then they fuse back into these lamellae. So we have a coupling of polymers going through three distinct phases, subject to different stresses, subject to different processes, a very rich environment. And this sets up what we call the kinetic trap, which cycles these populations where polymers accumulate faster than they degrade due to hydrolysis. So what we predict is that this forms what Carl Woese and George Fox called a progenote, the transitional unit that will carry protocells to complex living microbial cells. And it's this ignoble sludge, this gel aggregate. So let's put it all together. The big picture: hot spring hypothesis for the origin of life. We start with synthesis in the atmosphere and in the early solar system accretion, accumulation on land, which sets up our first chemistry, concentration, which allows membranes to form and things to get together. So that material might find itself into a cycled hot spring pool where our three-phase cycle can occur, generating more and more robust proto-life, proto-cells, which then, when washed down the channel into more dilute settings, have to start feeding from sunlight rather than energy from the vent or the hot spring feeding system of redox sources. So you get early life, which can divide within the context of the community. So progenote becomes microbial community. Then you get adaptation in an estuary environment to salt environments, and then colonization of the marine shore, which is abundantly exhibited in the fossil record. So here we have our Geyserite from the hot spring environment, our lacustrine stromatolites, and then we have the large marine stromatolites in the rock record. So this puts it all together. Implication number one for this is that there are two alternative paths for origin evolution of life. If you look at the origin of life on land, it's really thermodynamically more likely. because you have an origin in a hot spring setting. Photosynthesis can evolve directly in the hot spring environment, carrying microbes into independent lives, marine cyanobacteria, photosynthetic bacteria, all the way 3 billion years towards freshwater algae and land plants. And we now know that land plants have a freshwater origin, whereas a life beginning in the hydrothermal vent is thermodynamically uphill all the way, because as soon as you leave the vent environment for any protocells that might form there, they starve in the bulk of the ocean. They would have to instantly adapt to sunlight capture. They have to adapt to symbiotic fusion in a dispersing dilute environment, and then basically make their way into the marine algae environment. It's really, really quite a climb. It's quite a challenge for an origin of life deep in the oceans. Implication number 2, in the search for life on other worlds, we've got the discovery of a hot spring of the age of the ones discovered by our colleagues at Columbia Hills. And we predict that perhaps the first and last outpost for life on Mars would have been a hot spring. And as Mars lost its surface water, life would escape into the crust and be existing as halophilic bacteria. So hot spring outcrops or formations are very good places to look. And Perseverance will look for hot springs at in the Jezero crater toward the extended part of its mission. So there's some olivine silica that was found by Spirit when its wheel stopped working. That's definitely hot spring material. Here are these digitate silica structures found by Spirit. They're indicative of a hot spring on Mars. And lastly, we predict that for life to start, you need this kind of a dictum. You need this requirement. You need to have polymers with encapsulation and combinatorial selection. Polymers long enough to hold information and fold and have catalytic activity. And that aqueous carbon chemistry and cycling pools on subaerial volcanic landscapes is required for a life to start on Mars, but wouldn't exist in the ocean worlds, wouldn't exist deep in the hydrolyzing environments of these environments. So life can't start there, but it might still be habitable. So life could have been seeded there from elsewhere, but is unlikely to thermodynamically be able to start there. There's been a lot of public and scientific interest in this. Just yesterday on the BBC, here's our Scientific American article for the public in August 2017. And they did a wonderful job of the landscape of the cycling system. And you're all welcome to go to search for hot spring hypothesis for origin of life. You'll find an open access cover story from April of 2020 Astrobiology. It's took us about 4 or 5 years to produce this. It's the complete hypothesis. So take a look at that, and that's the image from Dave's first slide. If I have any more time, I want to describe new directions for you. So when you look at passive protocells going to active living cells, this is the great chasm we have to cross.
2 minutes there.
2 minutes, wonderful. So it's the chasm between physics and biology. And this chasm is non-trivial. And the prediction that we're making right now is that passively assembled protocells that might have a few functions, they can't live on their own. But when you aggregate them into a clump, into an aggregate mass, as we're showing would occur in the couple phases cycle, a reaction over here, a metabolic process over here, can diffuse products throughout this protocellular mass and start another reaction here and start another one here, and that it can set up a network effect. And we all know the power of networks. This is from the internet, what we're looking at right now. And so that the prediction is that this protocell aggregate, which we're naming the progenote, has the ability collectively to adapt and grow long before the individual members would have that ability, because they don't have the technology to divide themselves, et cetera, but the collective could. And so we're saying that this is the unit that carries, as Woes predicted, this is the unit that carries us to living cells. This unit has the following properties. It shapes probability by crowding polymers together, so things are more likely. And the roll of the dice there, and then the forms interconnecting networks, which I just explained. and allows a memory system to arise, templating, the templating of products, the first protogenetic activity. And that this cycling of these protocell populations around and around and around is what carries you toward the origin of life, or the emergence of the coherent individual living cell within this mass. And so some of the predictions we come up with recent work is that functional polymers may first arise co-located on membranes. They're not floating free in the lumen of the vesicle because it's too dilute. It's like the ocean. But if they're attached to membranes, this is work from University of Washington recently, they can get together. They're concentrated, attached to the membrane. That's a prediction. The aggregates of protocells represent the first primitive progenome. That's a prediction. You can start cycling. You could create progenotes with synthetic biology. Kate Animal has talked about this. Anna Wang has talked about this. And that other colleagues have shown that hierarchical reaction networks can emerge within systems like this when you subject the whole system to environmental stresses. And that we can get the first primitive genetic memory starting with templating in this system. So these are all future experiments. I'm working on a book together with several of you on this call and Dave that will be a cookbook for experiments on this. And lastly, just thanks and acknowledgements to all of our collaborating institutions. And I want to put a shout out to McMaster and Ben, they've just emerged as being major leaders in Origin's work, along with everyone else we've been working with. We've been working with them as long as anyone at this point. And thank you, Michael, for allotting this time for us.
Absolutely. Very good talk. I love this story. And the fact that you guys are pushing it in so many different directions I really do feel like there is a paradigm thing that is happening here. So it's very exciting. That said, we are running about 6 minutes behind